Please feel free to start, Sarah.
Okay. Hello, and thank you for joining us for the Old Dominion University REYES program. I'm pleased to introduce today's speaker, Professor Rachel Childs. Rachel Childs received her bachelor's and master's of science degrees from Virginia Commonwealth University. From there, she began teaching as an adjunct instructor at ODU while working full-time as a medical laboratory scientist at a nearby hospital. She is currently a clinical assistant professor at Old Dominion University within the medical laboratory science program, teaching hematology, urinalysis, and phlebotomy. Thank you so much for joining us today for her lecture, Where Does Our Blood Go? And I will turn it over to Rachel.
Sarah, and welcome everyone, whether you're attending live or you're attending in the future. I'm so excited to have this talk with you guys. We're going to be discussing where does our blood go? So what that entails would be, we're going to start from the very beginning of blood processing. So from the start where you get your sample collected, whether that's at an outpatient clinic or at a hospital, all the way to the end where you receive that lab result. Okay. Myself, as Sarah mentioned, I did get my degrees from Virginia Commonwealth University. And after graduation, I started working full-time in a hospital while working adjunct at ODU. And just recently, about a year ago, I kind of swapped those two positions. So now I work part-time, or what they call PRN, at the hospital. And then I work full-time here at ODU as a clinical assistant professor. About today. So first, we're going to provide an overview of blood. So what is blood? What are the components in blood? And what are the cells that are present in the blood? We'll also discuss sample processing. So we'll talk about the workflow from getting that sample collected all the way to receiving that test result. And then we'll review a little bit about what medical laboratory science is and what a medical laboratory profession scientists would do in the hospital laboratory. A bit about what your blood is, what it's made out of, what are the cells or components that are present. Blood. So everyone has about 5 to 6 liters of blood in their body at any given moment. So that blood is composed of cells and plasma. So within your cells, you have your red cells, you have your white blood cells, so your immune cells, and then you have your platelets. So the ratio between your cells and your plasma, which is more the liquid portion of where those cells are composed of, it's about at 50-50. So it's not exactly 50-50, it might be 45-55, it might be 55-45 reverse, but it's roughly of that 1-to-1 ratio of each. So that then your plasma, as I briefly mentioned, is just the liquid portion that contains all of those cells in the blood. So your plasma can contain proteins, lipids, vitamins, other things that are nutrients to your cells and tissues. Those are going to be floating within the blood as your blood travels through your blood vessels. Naturally, your blood will separate into those two components, your cells and your plasma. And by this picture here, this is the natural way that your blood will settle if left unattended for over 30 minutes to an hour. So naturally, your blood is going to want to settle where you're higher in weight. Proteins and cells are going to float to the bottom. So those are going to be where your red cells are going to be. In the middle, there's a very, very small layer called the buffy coat, which contains your white blood cells and your platelets. And we will talk about all of the different white cells that are present in your blood, as well as the functions of each of them. And lastly, at the top layer of your blood is your plasma. So that again contains water, proteins, vitamins, other solutes that are going to provide nutrients to your cells. Talk about those cells in your blood, so your red cells, your white blood cells, and your platelets. So first, your red cells, so more scientifically termed, they're known as erythrocytes. They are the most prevalent blood cells in your blood, of course. So where you see the red color, that comes from those red cells that are present in the blood. Known as a biconcave disc shape. So if you can see in this picture here how it has that concave or dip in the red cell, that is the normal shape of how your red cells should be in your blood. So one fun fact about your red blood cells are that they do not have a nucleus. So they don't have mitochondria, they don't have DNA in your red cells because they do not have a nucleus. And the function of them is to supply oxygen to your cells via a protein known as hemoglobin. So within that hemoglobin protein that is also within the red cell, you have oxygen molecules there. You also have iron that's present within the hemoglobin molecule. And you also have other proteins known as globin that are present in that one specific protein known as hemoglobin. Next, you have your platelets. They are very, very small blood cells within the blood. They maintain blood vessel integrity. So normally your platelets are just surveying your blood vessels, making sure there's no tear or blood vessel injury. However, their main function you'll only see that occur when there is a vessel injury or where there's a tear or you cut yourself. So that's when your platelets really take action. So during this process, your platelets are going to help form clots to prevent excess blood loss. So that's the main function that platelets play within your body. Red blood cell, your platelets do not have a nucleus, so there's no DNA within your platelets. Platelets are that some of them are stored in the spleen. So about 1/3 is stored in the spleen. So the purpose of that is because, say, if you had a massive accident or there was something that happened to your body where you lost a lot of blood, you have a storage component in that organ, the spleen, to provide the adequate amount of platelets to come to the rescue when there's excess blood loss. So blood, you don't have to wait for more platelets to be produced to come to that scene of the blood vessel injury or tear. You do have 1/3 that's stored there at any given moment in the spleen. Here. Okay, so the largest category of cells in your blood would be your white blood cells. They all play some type of role in the immune response. Okay. Antibodies to fight off infection. Some of them play a role in phagocytosis, which is the ingesting or engulfing of foreign material to kill it and then recycle any of its products. And then a role in allergic reaction. So you'll see some of these cells will be increased if you had a medication allergic reaction or if you had some type of hive or rash that you see in a specific area. 5 major types of white blood cells. It's divided into 2 major categories. You have your granulocytes, which are composed of granules. So the granules are all of these little dots that you see in these large white blood cells here. So those are going to be the granules, and that's how they get their name, the granulocytes. Then you have granulocytes, which are white blood cells that do not have granules scattered throughout the entire cytoplasm of the cell. The most common white blood cell is going to be your neutrophil. We see that in the highest frequency when we test our blood. So your neutrophil is going to be responsible for that phagocytosis function. So digesting and digesting foreign material. Your eosinophil and your basophil white blood cells are very, very small in number. So there's not that many that are present in normal blood. So your eosinophils are going to be responsible for allergic reactions as well as your basophils. To your agranulocyte category, your lymphocyte, that's going to be your second most commonly seen white blood cell in your blood. So your lymphocytes make the antibodies. And at least we have our monocytes, which is the third most common white blood cell that we see. And these monocytes are going to be also responsible for phagocytosis. Now that you have a brief overview or a general overview of what is in your blood, we're going to talk about how your blood goes from being collected in the lab or a hospital or outpatient setting all the way to receiving your results. We'll go through each of these 5 steps in detail. But as an overview, so first you have that sample that's being collected. So whether you're sitting in an outpatient setting or you're in the hospital, somebody's collecting that sample from you. Then you have the sample being transported to the lab where the testing will occur. Once the sample reaches the laboratory, us as medical laboratory scientists have to determine what specific department that sample needs to go. So that's where we designate the specific lab department that performs the test that your doctor ordered. Then we do analysis, so where the sample actually gets the testing done and you get a result. And then finally, that result needs to be entered into the patient's medical record. Sample collection. Samples that we can receive in the lab, I will say blood is the most common sample type that we have. And there's different specifics of blood that we can test as well. So we can take blood as a whole, so without separating out the components. We can test just the plasma. So when we talked about the separation of how our blood is, the plasma would be that top layer. And then we can also test similar to plasma. There's a few proteins that are missing from it. And we'll talk about that a little shortly. We can see bodily fluids, so whether that be urine, whether that be sputum, or whether it be other fluids within your joints. Then we can receive throat, from a wound infection, pretty much anything that can come out of your body, we can receive and we can do testing on it to get a result. So I don't know, most people don't know this, but the color of the tube for blood, those mean different things. So it's not just because we like to look at rainbow colors and it looks cool to see. So there's different components in these tubes that tell us just by looking at the color, what is in that tube. So the tube top color corresponds to what specific department or what anticoagulant or other substance that's in the tube. So sometimes these tubes, if you can see some of them, they have what's known as a gel barrier. So this yellow substance at the bottom of some of these tubes, that is what can separate out plasma versus serum. So sometimes we want the blood to clot, and then when that separates, we get. However, when we don't want the blood to clot, we want it to stay as a whole blood component when it naturally separates plasma. So depending on whether the test calls for plasma or serum, we'll determine whether we want a tube with a clot in it, or a tube with a gel barrier in it to induce a clot, or a tube that will contain anticoagulants or other components that prevent the blood from clotting. So that's a fun fact about tube collection. So there's other requirements that phlebotomist or whoever collects the sample needs to keep in mind when drawing blood or taking a swab from you, etc. Some tests that require the sample to be warm, so it needs to stay at body temperature. So that needs to be kept when a phlebotomist or whoever is collecting the sample is looking at the test that's ordered. Most samples need to stay at room temp. That's the most common temperature requirement, which is great. And we also have certain samples that need to be on ice when they are received in the lab. So by putting in ice, that prevents any type of metabolic process after it's collected. So some tests, we need to see what level a certain analyte is right at that body temperature level. So by freezing the sample, we kind of freeze those cells from performing metabolic functions, which we can then monitor at that specific time the sample was collected, how those cells are working. At least, there are some tests that require the patient to be fasting for whether it could be 6 hours, 12 hours, it really depends on the test. So it'll be something to keep in mind when it comes to getting your sample drawn, or if you're following the directions that your doctor has provided if you need to get your blood drawn. Now that the sample has been collected, it's time to bring that sample to the laboratory. So no matter what the sample is, whether it's blood, bodily fluid, urine, etc., it all needs to be collected in a biohazard bag. So that indicates to us that this is potentially infectious material. Whether it truly is or not, we treat all samples as if they were infectious, just to keep us safe and keep the people handling the samples safe as well. For different ways that we can receive the samples in the laboratory. So say if you're in a hospital setting, we can have either nurses, anyone drop the sample by hand delivering them to the laboratory. We also have what's known as a pneumatic tube system. So say if you're in a large hospital system and you're several floors away from the laboratory, if that hospital system has a tube system, they could just tube the system or tube the sample, to the laboratory. So they don't have to walk down there and drop the sample off. Collected at outpatient setting, or if the test is performed at a completely different hospital, we have lab couriers that would come and deliver the sample. Well, they'll pick it up from the said specific location and drop it off to the laboratory that the test is being performed. So there's mainly 3 different ways of transport that we can receive the samples. Now that the sample has reached the lab, it's reached the specific area or specific hospital that the test is performed, at the laboratory, we need to separate out which department it needs to go to or which department the test needs to be performed. So there are several different departments in the laboratory. Here I have just a few of the commonly seen ones at a given hospital. So we'll talk about the blood bank department, the chemistry department, hematology, urinalysis, microbiology, and molecular. First is the blood bank department. This is where we are going to determine patients' blood types. We're going to perform compatibility testing, so to make sure the specific donor blood that we receive from the Red Cross is compatible with that patient receiving that blood product. We also have antibodies that would make the donor red cell unit incompatible with that patient. So a lot of different testing on the donor cells as well as the patient cells. And after we've done all of our tests, we also need to determine which specific donor units are compatible with that patient. So we will match the donor unit to the patient and then set up what we call gated units to that specific patient. So if you are paired with a specific donor, that donor cell or that donor unit stays with you until you either it expires or the patient leaves the hospital. So we really do a good job of testing the samples, both the donor and the patient, to make sure your blood is compatible. The largest department for sure in any given laboratory, just because there's so many different tests that are performed under the chemistry department. So, but mostly your chemistry tests are going to determine your status of a specific organ. So we can do liver function tests, we can do kidney function tests, heart function tests. We can also do electrolytes, so like your sodium, your potassium, your chloride, just to make sure you're well hydrated, as well as all of those analytes are working properly and at their, proper levels. There's so many different tests that can be performed under chemistry. Section of the laboratory is just what I have the most experience in. So with the hematology laboratory, this is where we can assess those blood components that I mentioned earlier. So we can look at your white cells, we can look at your platelets, we can look at your red cells. So not only do we see are they there in normal quantities, we also assess the morphology of them. So if they don't have that biconcave shape of the red cells, that's something we would notate and notify the physician. So we give the numbers of those blood cells, so just how much of them are present in the blood, but we also assess the morphology of all of those cells as well. Microscope focus department. So a lot of times you will be under the microscope for most of your shift if you're working in that section of the laboratory. But it's fun because what you can do with hematology is identify or help diagnose leukemias, sickle cell anemias, as well as infection. So that list is also not all encompassing. There's so many different disorders that we can identify in the blood, but those are just a few of the most common ones that we see. This would be urinalysis. So there's many different components that we can assess in urine. So we can look at physical examination, so the color of it and the clarity. Sometimes just looking at those two different tests can tell us a lot about the urine itself. So normally it should be clear and yellow. However, sometimes we've seen urine of all different colors, as well as with the clarity. So normally you want your urine to be clear. However, cloudy urine can indicate certain versus others. What about urine just by physically analyzing the sample? Also do chemical tests on urine as well. We look at the pH of the urine, we look at the protein and the glucose content, et cetera, in the urine as well, to help us tell a little bit about what that patient may be experiencing. And then lastly, we can look at the urine under the microscope. So in this picture here, which is a picture of we can also see bacteria and other white blood cells that could be present. So normally we shouldn't see anything in the urine. However, if we do see something microscopically, that can indicate potentially infection, a UTI, yeast, etc. Identify just by looking at the urine, so we can assess kidney function, know whether the kidneys are functioning normally just by looking at certain chemical tests in the urine, as well as tell if the patient is adequately hydrated or not. So just by looking at the color of the urine, we can assess whether that person has drank enough water or may need to drink more water in order to be adequately hydrated. And last but not least, we can also identify diabetes. So if a sample has a lot of glucose in the urine, that patient most likely could be a diabetic patient or has a loss of kidney function as well by This is a really fun and interesting department to me as well. So what happens in this specific section of the laboratory is primarily we take whatever sample that we receive, whether that's a swab, that could be a wound culture, that also could be urine as well, if they want to look for specific organisms that are growing. So what we do is we have these agar plates, and then we're going to swab that sample onto the agar plate and watch and see what grows on those plates in roughly 2 to 5 days, depending on the type of sample. 5 days, we start with this normal blank agar plate. We streak the plate with whatever specimen or sample that we receive in this department. And then after that 2 to 5 days, we see different growth patterns depending on the organism that's present, depending on the specific agar plate that we use to plate the organism. So just by looking at the color and the size of those colonies that are formed can tell us a lot about the organisms that are present. Our growth patterns on these agar plates, we can identify the organisms causing disease or infection, as well as from the colonies formed, those colonies, to see whether they are either resistant to certain medication or they will be susceptible to certain medication. So after we have identified the organism, we perform other tests to identify to the physician or provider if that organism is resistant to certain medication or is susceptible. We pretty much are giving the physician a list of medications that should be treated with that patient in order to kill that organism that's causing the infection. So last but not least, we have the molecular department. This is an ever-growing department in the laboratory. With molecular departments in the lab, we can identify genetic disorders. We perform DNA and RNA testing on the samples, as well as COVID tests. So if you have done a PCR test or have gotten your nose swabbed for a PCR COVID test, that is most likely going to be performed at a hospital or a research laboratory where medical lab scientists are performing those tests. Molecular is that we can identify new mutations. So going back to how we have different strands that are becoming more prevalent, that's also something that can be identified by medical lab scientists as well. Now that the sample has reached the specific department, we're ready to perform the test that was ordered. Analysis. One thing before we can do any test is to ensure that that sample is of great quality. So we don't want to have a poor quality result because that sample was. So sometimes that may indicate that the sample needs to be rejected. Not because we're being picky about the samples that we receive, we really just want to make sure that we're doing the test properly and that that result is accurate for what's going on in that patient. So the first step would be ensuring the quality of that sample. And then the next step would be ensuring the instrument quality before we even perform any test on said sample or patient, et cetera. With instrument quality, a lot of that entails the maintenance of the instrument. So sometimes that could be something daily that's performed or weekly, monthly, et cetera. Sometimes that also entails quality control, or excuse me, we always want to make sure our instrument is performing at the level that it should be before we even run any patient test. And then lastly, we want to document for future reference just so that we have a record that the instrument at any given time during the day is performing at the level that it should. So all of that is involved before even a test is run on the instrument. Now that we've run our test, we've made sure the sample was of good quality, we made sure our instrument was working properly, we performed that test. Now it's ready. Now we're ready to give the provider the result for that patient. There's a lot more that is involved than just sending a result out to the medical record of that patient's chart. So some things to keep in mind: if there are critical values for a patient, we need to call those to the provider or nurse before we can even enter that result into the patient's chart. What the medical laboratory science professional does is call in criticals to make sure that the nurse or provider has that result even before the sample or before the result can be entered into the system. A lot of times when we receive or when the result gets entered in the patient's chart, it looks similar. Where test is performed, the result of the test, and then the reference range so that the provider can see whether that result is higher, normal, or lower than what it should be. So as an example, this patient here has a high white count, their sodium is low, and their glucose is high. There are many ways that a result can be entered into the system. The most common way would be known as auto verification, where if there's nothing wrong with the sample, we don't need to call any criticals, that result just automatically gets entered into the patient's medical record. There are times when we have to manually enter that result into the patient's chart, usually manipulation to the test. So if we needed to do a dilution, we had to do the twice because something happened with the instrument or the sample itself. So either repeating the sample, that might require manual entry of that result. Ways that results can be entered, they can be auto-verified, so it automatically goes into the chart, or you would have to manually enter that result into the patient's chart. Our sample from the blood being collected, getting transported to the lab, we've designated what department it needs to go to, we've performed our test, we've entered that result, or that result has been entered into the medical record of the patient's chart. We've done everything that we needed to do, and that blood has reached its final destination. A little talk or overview of what MLS or medical laboratory science is, as well as how to become a medical lab science professional. Year 4. So we definitely are looking for someone who loves science, of course. In medical lab science, as well as most healthcare professions, we're looking for detail-oriented individuals. So like I mentioned with the test analysis, we need to make sure that sample is of good quality. And it takes someone who is detail-oriented to sometimes pick out different things that could be potentially wrong with that sample where it would need to be rejected. Looking for detail-oriented individuals as well. Problem with instrument quality. So sometimes instruments, they break, they faulty. So before we can even do patient testing, we need to be problem solvers to figure out what is wrong with our instrument and get it back up and running so that our test can be performed at a high quality level. So we're looking for detail-oriented individuals. We're looking for problem solver individuals. And this last point, I will say it's not 100% the case, but with medical lab science, you're not interacting with the patient directly. You're mostly that person behind the scenes, giving the result, helping with patient care without directly interacting with the patient. For those of you who are introverted, or for friends that may want to help with patient care but don't necessarily feel comfortable seeing the patient one-on-one, this might be a great career field for you. Now, that doesn't necessarily mean if you are extroverted that you can't be a medical laboratory scientist. All gamuts of individuals who are medical laboratory scientists, but in general, this tends to be a field for So this degree is a bachelor's degree as well as an associate's degree. At ODU, we have the bachelor's degree option. So that's the one I'm going to talk about more at this time. So with ODU's program, it's a 2+2 program. So you do 2 years of your gen ed undergraduate coursework, and then you do 2 years that are specific to the program. So it is a program that you have to do a secondary. I mentioned this is a bachelor's degree, but it is something that you can also get your associates in. At ODU, we have about 1.5 years of coursework specifically in the program, and then you do 1 to 2 semesters of clinic rotations. So that's where we actually send you into those hospitals and you get to see one-on-one firsthand what your medical lab scientists are doing. And you also might get to help out reporting results, with getting the samples ready for testing, et cetera. So that's really where the students get to see firsthand, do I really like this? Do I enjoy this? Is this something I really want to do? Then lastly, after graduation, you do have to be certified as a medical lab scientist professional in order to practice in. So it is an additional board exam that you have to take after graduation in order to be certified. After you perform all of those steps, you are ready to work in anywhere in the hospital, as well as other outpatient settings. Do we work? So most commonly we'll work in hospital laboratories, but we can work in research facilities, clinics, urgent care. So for example, those CHKD outpatient clinics, as well as patient first, those people who your blood and performing the test are medical laboratory scientists. So that's public health laboratories. So think of the CDC or the WHO. All of the ones that are doing laboratory testing in those facilities are most likely medical laboratory scientists. That is all that I have for you today. Again, for attending, and I'm open to any questions that you may have.
I don't think there's any questions right now, but I know that the viewership will increase with the recording. So there may be some questions that come to you after the fact.
So yeah, feel free, Sarah, to share my email address with anyone that's interested or has any questions.
Thank you for your lecture today.
It went shorter than I expected. For listening.
Have a good day.
You too. Bye.